All right, hello everyone. Today's topic is on ECG, or if you're old school, EKG, electrocardiograms. And it's trying to understand this structure that we've got that I've drawn up here. We can label these peaks and valleys. A P wave shown first. This is going to be followed by what's called the QRS complex, with each peak being labeled. And then we finish up with what's called the T wave. Now, it's easy enough to define what these things actually represent. We'd also like to show how they're formed. But first, let's do the easy part. Let's show what they represent. Let's start with the P wave. The P wave simply represents atrial depolarization. We return then to baseline, and then there's a bit of a gap there. And then we initiate the QRS complex. And QRS is all about ventricular depolarization. We once again return to zero, to baseline. And that leaves us with the T wave, which represents ventricular repolarization. Now let's see if we can understand all of these in the context of the electrical conduction pathways of the heart. So if we were to look at the heart divided up into two sides, of course the right side is driving pulmonary circulation, the left pump is driving systemic circulation. Each side has the atrium, which receives the input of blood, and then the ventricle, which is my primary pump. We can trace out the electrical conduction pathways, which start with the SA node in the top corner of the right atrium, where SA stands for sinoatrial. Of course, from our previous discussions, you would recognize that this is a cluster of cells that are autorhythmic cells or pacemaker cells that utilize funny channels, HCN channels, to promote their own self-stimulation. And the SA node beats with an autorhythmicity of about 80 beats per minute on average. Now, where does it go? Well, we got some rapid conduction pathways. Of course, we got Bachman's bundle. We need to take this over to the left side of the heart. But primarily, we're diving down here to our next stop, which is the AV node. And if the purpose of the SA node was pacemaker, the purpose of the AV node is simply to slow everything down. Now, this actually represents a very interesting topic, because as we talk about, the rapid conduction pathways between these two nodes, we've got to get the signal there kind of quickly, but then we want to slow everything down. And from a physical point of view, slowing it down is important because we've got to give the atria time to physically contract, the muscle to contract, to move that blood, to squeeze that blood into the ventricles before we tell the ventricles themselves to contract and squeeze it out. And so slowing it down here is a critical step in the process. I'd like to take just a second to, in an oversimplified sort of way, kind of show you how that works. Remember, these electrical conduction pathways are driven by gap junctions. Let's take a second to look at that and understand what that actually means. Let's start with a cell here. It's just a square, but let's say that it's also surrounded by 4 other cells. And these cells are all connected by gap junctions. Remember, these gap junctions are essentially just little tunnels or tubes that connect these cells so that they can freely share cytoplasm and any dissolved solute small enough to pass through these channels. So the white cell in the middle, it's this autorhythmic cell, right? These funny channels open below threshold, sodium starts coming in, we hit threshold, and we have voltage-gated calcium channels opening. So this cell, if we kind of zoom in and look at it, starts to fill with calcium. The entire principle of an electrical synapse, as opposed to a chemical synapse, I don't have to deal with neurotransmitter. It's all about ions traveling through these gap junctions. So calcium comes in here, calcium comes in here, calcium comes in here, calcium comes in here. And as these blue cells fill with calcium, the calcium in and of itself is sufficient to help that cell depolarize to reach threshold. Right? These are now the cardiomyocytes. They're way down. They've got a resting membrane potential of minus 90. But as this calcium comes in, they start to approach threshold. At threshold, what happens? Voltage-gated sodium channels open. So now through these voltage-gated channels, we've got sodium rushing in. Of course, this is an oversimplified view. I'm just drawing one cell. But what's that sodium going to do? As this cell that it enters is depolarizing and going through this action potential, some of that sodium is going to leak right through gap junctions into the next cell and cause this cell to fire an action potential. And so we propagate it cell by cell as each neighbor essentially fires, but in that firing is also sharing ions with its neighboring cell, telling the neighboring cell to fire in a chain reaction sort of event. Well, how can we slow this down, or how can we make it faster? Without getting into too much detail, I just want you to think about size for a second. What would happen if all of a sudden we had just a bunch of small cells, each one taking its turn going through the process? I hope you can appreciate that with the decreased size, it's going to take longer for each one to fire and then trigger an action potential of its neighbor. And so a lot of what we see in the AV node is just a bunch of smaller cells that have slowed this action potential, this electrical transduction down, as we wait for the atria to physically contract. And then as we want to speed up again, what do we see? Bigger cells. So we're going to be slow through here, but we're going to be fast through here. A lot of it, and this is again an oversimplification, but a lot of it depending on the actual size of the cell. All right, let's go back and let's finish the electrical conduction pathways from the AV node. We then travel through the bundle of His. This divides up into bundle branches, with each bundle branch traveling toward the apex of the heart, where we finally hit our Purkinje fibers that start at the apex and then spread laterally upward up the walls of the ventricles. And we see this on both sides. And just as a review, nearly everything that we've got drawn up here in white is populated by autorhythmic cells, right? The SA node, the AV node, the bundle branches, the Purkinje fibers, these are all autorhythmic cells, each with their own autorhythmic rates. The reason the SA node is the primary leader of this is simply because it has the fastest rate. So the SA node has an inherent autorhythmic beat of about 80 beats per minute. The AV node, on the other hand, has an inherent beat of around 40 beats per minute, while the Purkinje fibers around 20 beats per minute. In this way, if the SA node fails, the AV node will then take over. Of course, 40 beats per minute, you're going to see a rapid loss of blood pressure. The person is going to probably pass out, but they'll come to, and beating at 40 beats per minute, I'm not going to go out and run a marathon anytime soon, but it's certainly sufficient to give me enough time to get to the hospital where I can receive medical help. And if it is indeed failure of the SA node, then they're going to schedule me for surgery in a few days, and they're going to put in a pacemaker that will use electricity to take over and artificially stimulate this process. All right, now that we've got that mapped out, our next job is to understand how this pathway from the SA node to the AV node, down the bundle branches and up through the Purkinje fibers, gives us this EKG reading that we've got over here with the PQRST stuff. And in order to understand that, there's a couple of things that we have to define. First of all, the electrocardiogram is a machine that's based on reading dipoles. A dipole is simply separation of charge, where I have equal and opposite charges across any given distance. Let's provide just a quick review of resting membrane potentials and action potentials and how that might establish a dipole in these cardiac cells. So let's go back to a square cell. Resting membrane potential. We run our sodium-potassium pump that creates gradients, and then we increase potassium permeability, and as potassium leaves, the cell gets negative. So inside the cell, we have negative charges, more so than outside. Of course, what that means is that outside we have positive charges. For every positive, there's going to be a negative. We're for the most part electrically balanced, but we've actually separated these charges across a cell membrane. Does that represent a dipole? Yes, it does, but it's not the dipole that we're interested in. It's not a dipole that the EKG can actually see. This machine doesn't look inside cells. It only sees what's outside. And right now, what's outside? All we see is positives. So let's simplify this further and let's say, okay, this cell is negative on the inside and it's positive on the outside. Remember, especially as we're thinking about these conduction pathways, let's say this cluster of cells starts with the SA node and ends at the AV node, right? We've got a directionality to how this electrical conduction is taking place. But before any of it happens, at rest, all the cells are negative, and outside of the cells, That's all positive. But let's think about the changes that take place as the heart contracts. The very first thing that's going to happen right there in the SA node is going to initiate an action potential. And what is that going to do? We're going to open up voltage-gated calcium channels. Calcium is going to flood inside the cell, and that's going to change the inside positive, and the outside is going to be negative relative to those charges. Now we can see the dipole that we're interested in. The dipole is right there, where we have a separation of charge, a positive next to a negative. Both of these charges, remember, they're outside the cell. And that is something the machine can read. That's something that it can identify and see. So with that in mind, let's just go through a quick review of how this electrocardiogram works. First of all, we need what are called leads. And on the simplest of EKGs, we've got a 3-lead system. On more complex ones, typically if you're going to go in and get a good readout, they're going to give you 12 different leads. What I'm going to show you is just 2 leads to establish the principle that we're looking at. Where on one end we have a negative lead, and on the other end we have a positive lead. These are physical wires actually attached to the skin in specific locations that are going to give the machine the chance to read the dipole moments established by these dipoles. Now let's just briefly make a list of fundamentals regarding how this machine's going to work. Number 1. Dipoles running parallel to the lead axis will be strongest, whereas dipoles running perpendicular to the lead axis will not be seen. What does this mean? Well, it means what we need to think about is how this might look with vectors. And I don't want to get too much into vector math, but vectors are super useful because they contain both direction and magnitude. To illustrate, let's give some quick examples. Again, let's say there's my negative lead, there's my positive lead, and let's say my dipole arrow looks like that. That's going to be a very strong deflection. But in another instance, let's say the dipole arrow looks like that. Because it's completely perpendicular, the machine can't read it. There's no deflection there. In the third scenario, the dipole arrow looks like that. Well, now we have to start thinking about the vector components of that arrow. If we look at our axes, we've got our perpendicular axis and we've got our parallel axis. And the arrow that we see in Y is really made up of 2 different arrows. It's made up of that arrow on the Y-axis, and it's made of that arrow on the X-axis. What the machine reads is what's parallel to the lead axis. It will read this bottom arrow. Now, just to highlight why that's important, let's do another scenario. Again, set up our perpendicular axis and our parallel axis, and another arrow that say looks like that. Most of this arrow is actually perpendicular, so the full magnitude, the strength of this is going to be lost. But are we going to see something? Yes, there still is a little component right there, not very big, but a little bit that is parallel to the lead axis. And so it's something that's going to be visible on the machine, but it's going to be weak simply because the vast majority of it is perpendicular to the axis, but a little bit is parallel. So the dipole parallel to lead axis is the strongest. Number 2, the dipole arrow won't always go in the direction of the depolarizing event. In other words, the dipole does not equal the depolarizing direction. And we'll show you why this is important. Let's come up and let's look at our example up here. The easiest way to think about this, let's again come up and let's look at our dipole that we've circled up here, where the positive is next to the negative. What we want to do is we want to identify the dipole, and we want to draw our own dipole arrow. This arrow down here is not the dipole arrow. This arrow shows the direction of the movement of this electrical conduction. The dipole arrow we want to always draw pointing toward the positive end of the dipole. Now I get that that's not how they really do it in physics, but it makes it easy for us to understand how to do it here. Why is that important? Because if I take this arrow that I just drew, this pink arrow here, and I ask myself, Is it pointed to the positive lead or is it pointed to the negative lead? In this case, it's pointed to the positive lead. That tells me that this will be a positive deflection. But let's look at a different scenario. For example, what if we just change the leads? What if we put the positive lead on the left side and the negative lead on the right side? Remember, these are just wires adhered to the skin. We switch them around, and now what do we see? We still draw our dipole arrow pointed to the positive end of the dipole right there, but now it's pointed to the negative lead. That would give us a negative deflection. Let's give another scenario. We put our leads back the way they were, but let's change the charge of some of these cells. In this scenario, we're all negative on the outside except for that far left cell. We do the same process. We identify our dipole. It's right there where I have a positive next to a negative. I'm going to draw my dipole arrow pointed toward the positive end of the dipole. Recognize, my dipole arrow is not going in the direction of this event, right? We've got this arrow down here. It's showing the direction of the event. But the dipole is just a static snapshot of the charge right at that given time. And right now, the dipole arrow is pointed toward the negative lead. This would be a negative deflection on my EKG. Okay, the third and final rule is the simplest of them, and that is maximal deflection will occur at the midway point of the conduction pathway. Now, what does that mean? Well, now we go back and we look at our directional arrow. This tells me that I started here, that I'm going to end over here at the AV node. Let's go back to our initial charges. I'm at rest. I'm ready to go. The first one fires. I see my dipole. I recognize the dipole arrow is pointed toward the positive lead. It's a positive deflection, but I'm not very far along the way, right? In my journey, represented by this arrow here, I'm only about that far along. This is going to give me a positive deflection, but the deflection won't be as strong as when it is at midway. So let's continue our journey. See, now I'm halfway, even a little bit more than halfway. What do I see? Right there, that's where I see my positive deflection. So if I was drawing, because this represents the SA node to the AV node, this is my P wave, what have I seen? I started at 0. I started my positive deflection, but now I've reached the peak. Of course, when the next one fires, I'm past halfway, so it's not as strong of a deflection. I'm starting to come down. And of course, now everything has depolarized. There's negatives on the outside all across the board. There's no more dipole. So where do I find myself? I find myself back at baseline, back at 0, no deflection. We've just created the P wave. We're going to continue in a second to map all of this out and show exactly how to create the QRS wave and the T wave and all of that. But I just wanted to take a minute to review what we have with our understanding of how the EKG works. So the EKG itself, the reading, it's made up of a P wave, a QRS complex, and a T wave that we've defined as atrial depolarization, ventricular depolarization, then ventricular repolarization. We remind ourselves that all of these cells are connected through gap junctions, these electrical synapses, and it's the ions rushing into one cell during an action potential that leak through these gap junctions into neighboring cells, triggering a chain reaction. And we can regulate the speed of this by the size of the cell. We then looked at the electrical conduction pathways of the heart. We start with the SA node with a native beat of about 80 beats per minute. We've got rapid conduction pathways to the AV node, where it works to slow everything down so that the heart actually has time to physically contract the atria. Then we speed it up again through the bundle of His. We divide into bundle branches, which spread rapidly down the septum, and then we hit the Purkinje fibers, beginning at the apex of the heart and working laterally and upward, squeezing the blood, if I'm on the right side, into the pulmonary trunk, if I'm on the left side, into the aorta. The machine sees all of that conduction pathway by identifying dipoles, which is just a separation of charge. And the rules of reading dipoles were as follows. The dipole parallel to the lead axis is the strongest. the dipole direction does not equal the depolarizing direction. It might, but it might not. And then maximal deflection occurs halfway between the beginning and the end of the journey.